In 2021, the Oncology Center of Excellence of the FDA launched Project Optimus to improve dose selections in early oncology drug development trials. They had observed drug approvals that resulted in excessive toxicity and wanted to increase the accuracy of drug dose selection in the process before approval. The three goals the FDA published on their website were to communicate the expectation, to provide frequent and early interactions, and to generally develop strategies to improve drug dose selection. In 2024, the FDA published a guidance document to explain further. The document has many thoughts and directions and advice, but very few concrete rules that could be followed or complied with. One of the general principles is to move from MTD to OBD. MTD stands for the maximal tolerated dose, and that is the old oncology way of finding the right dose. The drug doesn't cure everybody. We go up as high as we can to cure as many as we can. That highest dose is the maximal tolerated dose, and this is where we try to get away from with the Project Optimus. Optimal biological dose is a concept believing that there is a dose that is lower than the maximal tolerated dose and equally effective or maybe even more effective, and that needs to be found. That is the concept. And with that, they recommend a number of things, including efficacy and pharmacodynamic components in the dose escalation, multiple doses in the expansion cohorts to be compared by randomization and blinding, even in phase 1 study. Model-informed drug development is a big piece, and the word model appears many times in the document. To make all that happen, they offer early interactions. Now, that is the one and only recommendation that stands with all my calls among my friend drug developers. Everybody has some recommendations how to handle Project Optimus, but the only one that everybody enforces is follow the early interactions, because everything else is so unconcrete, you just need to talk it through. The document also has other guidelines which are standard and have been there before. It is just unusual to see this sometimes in phase 1 studies. That includes PK/PD, pharmacokinetic, pharmacodynamic, in particular with modeling. That includes patient-reported outcomes, the food effect to be analyzed early, and then they describe endpoints of efficacy, such as progression-free survival that has typically not been seen in early oncology trials. This is an example for a typical early oncology trial of the old way how we have done it prior to Project Optimus. There was a dose escalation component which may be guided through the 3+3 design, or more modern, typically through the U-BOIN design. And it looked at safety mainly. Pharmacokinetic was also assessed. Once we knew how high you can go with the dose, once the MTD was determined, we determined a recommended phase 2 dose, which may or may not be the same as the MTD, and enrolled another 10 to 20 patients to learn more about the drug and maybe a little bit about the efficacy. This is a new study design, as it may happen since Project Optimus. We still have a dose escalation. Now, that may still be guided by U-BOIN design. There are other designs, they come later in the video, but the most important component is then, once there is somewhat of an understanding of the dose and the final doses are to be selected, we don't run an expansion with 1 dose, we run an expansion with 3 doses, or at least 2. And the allocation of patients to those expansion doses is asked for to be done by randomization. That is the new thing of Project Optimus. These were the essentials of Project Optimus. If you want to learn more, stay on the video for a few more minutes. You have dialed into a video teaching series for oncology drug developers. In the following, we will give more details about the information from the FDA publications, address why people are concerned with Project Optimus, and what can be done to handle it. In the end, I will allow my own personal opinion and finally thank those that helped with making this video and display the conflict of interest. My name is Johannes Wolff. This is recorded in March 2025. This video is also the result of a vote among the audience for those teaching videos conducted in LinkedIn. Almost half of the votes went to clinical development videos, and the example was Project Optimus. This video, we will carry on with further clinical development videos and possibly then also do the acronyms series. Before we go into further details of Project Optimus, let's go to the borders of it. What is inside of scope and what is outside of scope? Well, within the document that the FDA has published, they listed a number of things that are out of scope. Among those are radiopharmaceuticals, cellular and gene therapy, oncolytic viruses, microbiota, cancer vaccines. They also list pediatric drug development. And then finally, they also list that the selection of the starting dose wasn't meant to be part of the project. Use the old guidance for that. By the nature where that comes from, outside of oncology is out of scope, because this is from a part of the FDA that is relevant for oncology. That means benign hematology, sickle cell anemia, does not need to worry about Project Optimus. There are some borderline indications where it is questionable, such as hemophagocytic lymphohistiocytosis. By definition, this is an FDA project, so it is for the US. If you're developing a drug outside of the US, you need to follow those guidelines. Project Optimus has no authority there. And then finally, inside the US, also never spoken out, it appears that investigator-initiated trials are less affected than sponsored pharmaceutical trials. There is a teaching video that was provided by the FDA in one of their events to explain how they see Project Optimus, and this comes as close as it can come to really understand what the FDA means with this. I really recommend watching it. This is a really important slide taken from the FDA video from Dr. Booth's presentation. Here he explains the concept, how we think about cytotoxic chemotherapy versus targeted therapy, and why the concept should change from maximal tolerated dose to optimal biological dose. So this is the world that I was trained in as a young doctor. We had chemotherapeutic drugs that were effective and toxic, and we knew when you give too little, you will not cure your patient. If you give more when it starts hurting, you will cure some, and if you give more, you will cure even more patients. That was very clear. It was also very clear that these drugs were horribly toxic, and the more you gave, the more you hurted your patient. So the idea was, given that efficacy here means life or death, we go as high as we can. And we understand that we are overdosing some patients, because some patients were probably cured with lower doses, but we didn't know which one. Because efficacy in oncology is a measure that it can be determined only much later than toxicity. Efficacy means the cancer is gone and then doesn't come back. You need a year of observation to see that. Toxicity you see immediately. So doses were determined by toxicity. Go as high as you can and then determine efficacy later and modify the dose if necessary. The modern thinking is somewhat different. We have much cleaner drugs now, drugs that bind to very few or really only one site in the cell. And if that is the case, and if that site is really relevant for cancer cell survival, you bind it, you inhibit, the cancer cell dies. And if you give more, you will have a few more cancer cells dying, but not much more, because the receptor may be saturated, and then going higher just doesn't bind more receptor. They're already all bound. In contrast, toxicity is typically very low in those doses where the drug just starts to target its target, just to approach its target. Yet when you go higher, then the drug does reach other things, approach other binds, other places, and then toxicity of various kinds come into play, and the higher you go, the more you have of that toxicity. If this is the case, you would wonder if going to the highest toxicity is a good idea. Shouldn't we go just as high until we reach maximal efficacy? Now, I promise to give my opinions only in the end. I can't hold on to. I have to say that this is a very theoretical graph, which you can produce in cell culture and maybe in enzyme kinetics, but I have yet to see something equivalent like that in humans. This was a very high-level summarizing slide of that presentation saying dose optimization, do not use MTD, and maximize the general knowledge for drug development. This is another screenshot from the same FDA presentation, and it gives sort of the summary of recommendations, what you should do for dose optimization, the questions that the clinical developers have. What do I do here? So their first point is go away from MTD. The maximal tolerated dose is not the way to go. Look at efficacy as well. Consider non-clinical data. That's not really new. We have always done that, but they reinforce that because they want sharper dose selection. They also look at PK quite a lot. There are a lot of pharmacologists in Project Optimus, and they look at PK pharmacokinetic and PD pharmacodynamic relationship. That those are biomarkers you measure in blood, for instance, to see that the cancer is shrinking or at least the target is reached. They very much make a point many times about modeling and simulation. They think that that will help drug development in the future a lot, and there is a whole big project MIDD in the FDA to help with that. They recommend that multiple dose levels are to be tested. They recommend that those dose levels are randomized and that they are compared before a final selection is made for doses in later oncology trials. This is a slide taken from the second presentation of the same video. This is mainly about modeling and shows the plethora of how models can help in various parts of drug development. We are coming to the document, the publication of the FDA, the guidance to the industry how to optimize the dosage, published in 2024. This document starts with an introduction that is quite similar to the videos where they explain why to go away from maximal tolerated dose, and then it comes to dose optimization recommendations. The first pages are still relatively vague. They talk about dose exposure relationships. Again, modeling comes up a lot in this document, but they also say the program depends on a variety of factors, which means concrete recommendations, things you have to do, are not really in there. They recommend the Oncology Dosing Toolkit, which is another website you can pull up. The FDA document carries on with further industry guidance for clinical pharmacokinetics, pharmacodynamics, and pharmacogenomics. Most of the things you can read there are not particularly new in drug development, but it is new for some of them to appear that early in early drug development. So for PK pharmacokinetic, they talked about linearity, absorption, distribution. Those are all the old terms. But then they also talk about population PK. That means knowing the pharmacokinetic of various people and put the people then together as aggregate data. That's something where you need a lot of data, and it typically happens later than in early oncology trials. They talk about dose exposure response relationships, and that has always been the core of what we have been doing, but we have been focusing in early trials on safety. Now, they say, focus on efficacy as well, and I think that's a good idea. They talk about the analysis plan for pharmacodynamic and pharmacogenomics. Now, the whole document is very heavy on pharmacology, and they believe that pharmacodynamics will help us find the right dose. Pharmacodynamics means you draw something from blood, which may indicate that the drug has reached the target, has target binding, target engagement, or even beyond that, you have a tumor marker that tells you how much tumor is in the body. Obviously, those are very helpful if they are valid, if they actually do show how much tumor is in the body, because they kind of substitute for imaging that can only be done once every couple of weeks, and you can draw blood in contrast weekly and get a very much better view of the dynamics of it. They talk about semi-mechanistic and mechanistic modeling. Now, modeling, pharmacokinetic modeling, has always been part of early oncology trials. This was done in typical one compartment or maybe two compartment models. They are asking for more complex models. Semi-mechanistic and mechanistic modeling means that you include biological phenomena such as the liver, the cellular membrane, the cancer cell being different from the normal cell, and then use what you learn from there to find a dose that is least toxic and most effective. They talk about intra-patient dose escalation and de-escalation. This was a component that was almost banned for a couple of years in drug development. This is how we started before there was a popular 3+3 design or U-BOIN design as it is now modern. We gave a very small dose to a patient, and then we gave more and more and more as the cancer didn't take over and wasn't faster than our finding of the individual dose, then we found the right dose and could fight the cancer. That is enormously difficult to analyze over multiple patients, and it is really an approach that was done in individual patient treatment experiments. And it was not very popular in drug development. The FDA is asking us to reconsider and put intra-patient dose escalation and de-escalations back in our protocols. They talk about the food effect. Now, food can have an effect, in particular on large molecules when they are taken orally. The resorption can be very different if it's taken with food or without food, and before you know this, then it's kind of random, depending on what the patient eat, not eat. There is a tradition to do food effect studies, but we typically do them a little later, not before we know if the drug is doing anything to cancer. And then finally, there is a recommendation to look at the interaction of concomitant medications, obviously also a very interesting topic, and given modern computer technology, not that hard to analyze. Further in the document, the FDA gives advice for trial design, and in particular to compare multiple dosages, which is in the views of many the core of Project Optimus. Now, they advise to look at products with similar classes, the so-called class effects. That's nothing new. We've always done that. They advise model-informed and model-based designs. Now, we heard that multiple times. We will keep hearing it. It is mentioned many times in that document. They talk about backfill cohorts. Backfill cohorts is something that comes out of 3+3 design, when a cohort was declared where it was cleared when you moved up to the next dose. And then when you finally reached MTD, they say, go back to the lower doses, fill a few more patients in, and they call that backfill cohorts. There is a lot of questioning about that and the ethics of that. They talked about randomization and blinding, in particular for these multiple doses. Now, know that in the old rules of statistics, we have always done blinding, but we have put behind the randomization and blinding patient number calculations based on power. We wanted to do a clinical trial where we're blind, where we don't see what's happening, at least to the effect that in the end there is a statistically meaningful outcome, or there can be. So that led to patient number calculations, power calculations, and that leads to very large patient numbers, as they are typical for phase 3. Using the randomization method in trials that are much smaller than that, and that for certain are underpowered in the traditional statistical thinking, is something that requires some believing and some convincing. I have seen many statisticians shake their head and not particularly following that idea of randomizing. without power calculations. Then they ask for a sufficient assessment of safety and at each dose. Among the efficacy measures, they mention overall response rate. That is the classical oncology early development efficacy endpoint, but they also mention progression-free survival. And that's an unusual one for early development. I personally don't see a problem with this. We could do this. We could have done that a long time ago. They talk about adaptive design and crossover. Adaptive design means the study sort of changes while it is ongoing. There are decision points that depend on incoming data. The data come in and then you make decisions such as we combine the drug with drug A or drug B, or we use dose level 1, dose level 2, dose level 3, and different schedules and these kind of things based upon data that come in from the study. Then the study design is adapted to those data. That's called adaptive design. Crossover is a phenomenon from randomizations where you allow a patient that had been randomized to one arm when that arm doesn't work to move over to the other arm. The design allows to use overall response as endpoint, but the endpoint overall survival disappears when you allow a crossover. The document continues to talk then about safety and tolerability, but most of the things you find in there are the old things. And don't forget, they're not changing anything on safety. The safety analysis is somewhat the same, and therefore also the things you read in there, such as stopping rules, are not really new. Among the things you can see there is the unusual thing of evaluate titration. That means intra-patient dose escalation, for instance, is another word for titration. And they come to that idea with the experience of immuno-oncology, the cytokine release syndrome, which made that necessary. You also find PROs in there, patient-reported outcomes. That's also a component that we typically didn't use in early oncology trials. We use that in later trials. It means you ask the patient how they're doing, typically with very specified questionnaires, and analyze that then later. And obviously that helps when you need to find out if a tumor shrinkage that you measured in an MRI actually means something for the patient. Is the patient better? But those type of questions have traditionally been our questions later. And then they recommend to engage the patients. What they mean here is engage patients in the development of protocols. The document contains further recommendation for drug formulation, and it is obviously important if you want to test multiple drug levels that you actually have various dose strength available. And then the document has a chapter about what to do once you have the first idea and talks about subsequent indications, different cancers to treat, and usages. And the usages include multiple combinations, and it has some recommendations for that as well. So this all sounds very reasonable. Why would anybody be opposed to improvement of drug development methods? Why would anybody be against knowing better which dose is right for patients? Why would anybody fear Project Optimus? Well, I've put the reasons together. People do, in fact, fear Project Optimus, are bothered by it, and here are the reasons. Number one, that's the one you get from practicing physicians, and I would think probably also from patients, although I haven't asked any patients for this one. The critique is too many patients are underdosed. Project Optimus asks for different dose levels lower than the maximal tolerable dose. That's the whole concept of it. And that would mean that if those doses are not enough, then the cancer of those patients will grow and patients will not survive because the dose was underdosed. Now, you can say this is all clinical research that really isn't standard of care, and people shouldn't expect to have their cancer cured when they enroll in clinical trials. That is an unreasonable expectation, but it really isn't. That is indeed why people enroll in clinical trials, and also by law we cannot tell them that we think this is going to work. Everybody does that. Everybody thinks I'm going to enroll because it works. Thus, giving people then doses that you know are lower than what they can tolerate and may have lower effect on the cancer is a questionable thing. The second critique comes from drug developers, from pharmaceutical companies. And they say the phase 1 studies have become too large since Project Optimus. Remember, before 3+3 design, dose expansion, 15 patients. After 40 patients, we knew the maximal tolerated dose, recommended phase 2 dose. We had the toxicity profile squared away, and we had some idea if the drug is working and how well. Now, you do phase 1 studies and you start with 100 patients and more. Those are very expensive trials, and it is difficult to convince the sponsors to give their money for these trials before they know if there is a drug. If there is, well, there is a drug is a term from drug development. It means, do I have a drug? When the drug comes out of preclinical development, some of them make it, very few make it, most of them don't make it, and you want to know if you have one in your hand that can make it, and for that, most importantly, you want to know human efficacy. We have done these small trials before we knew human efficacy, and did the larger investment then when we knew that some of the cancers had shrunk. Now we're asking the sponsors to invest into clinical trials before we know that the drug is a drug. It's difficult. I have seen people call Project Optimus a company killer or killer of drugs because drugs are just not tested anymore. That's too expensive. A third critique is more of a academic critique. The drug dose differences recommended in Project Optimus are too large, and it kind of goes together with the patient numbers are too small. With so small patient numbers as you have it in early drug development, it is very difficult to show differences between dose levels because the differences are small, and small patient numbers and small differences, you just see nothing. Thus, Project Optimus recommends relatively large differences of doses between the different cohorts. In chemotherapy, when you adjusted doses because they were too toxic, we dropped the dose from a cycle that was too toxic to the patient to the next cycle when we wanted to give the same treatment again by 25% or maybe 30%, and that worked. The toxicity was less. Trust an old chemotherapy guy. Here, in Project Optimus, we're dropping them by 50%. We have factor 2 or factor 4 of dose between the different dose levels. That can only mean that one of the doses is really way off. And it will not really give you the fine tuning of the drug dose, which is in orders of 25%. The doses are just too large in difference. Same thing with the patient number too small. It's kind of an argument in the same direction. Because the patient numbers are small, it is really hard to show statistically significant differences between the doses, and that means the differences remain uncertain even after those 100 patient studies in phase 1. There is a critique point coming mainly from clinicians that this whole thing overvalues pharmacodynamic, and they think the real answer to drug doses needs to come from human toxicity and human efficacy. And then finally, the old critique of there is not a single dose, the whole concept ignores the differences between different individual bodies which determine toxicity of a drug, and differences between cancer which determines efficacy of the drug. In the past, we have always invested a number of resources into those individual differences, and those investments came after we had determined the rough maximal tolerated dose. Now we are investing all our energy and all our money into trying to find a maximal tolerated dose, completely ignoring the large differences between different people. Those were the critiques I heard. And the next part of the video will show you what I think we can learn how to handle all that. So this is a paper from Dr. Subbiah. He is in Sarah Cannon, the leader of the phase 1 group, and he describes the Project Optimus to clinicians and what to do about it. Now, this is mainly a paper indicating how different studies are and trying to help to not be too afraid of Project Optimus. The figure here places the dose optimization after the phase 1 trials. That's not the one and only way how it can be done, but it is interesting that from a very, very large clinician group, that is sort of their view. Now, this group, the Sarah Cannon team, has analyzed the changes of clinical trials since Project Optimus has come around. Their results are not published yet, they're still classified. So I can't give you the details, but I'm going to say two things. One is, by the time you see this video, the data may be published. Just Google Subbiah and see if it is there. And the second thing is, what that paper will show is that there were, in fact, meaningful differences in clinical trial designs since Project Optimus. So Project Optimus had an impact. And that also means you cannot ignore it. Now, our colleagues from biostatistics outside of the FDA, from academia, have weighed in to the problem of Project Optimus of finding the right dose. Among the papers I read for this video, this is the one I am recommending. It's published in 2024. It comes out of MD Anderson, and the first author is the same person who received the letter from the FDA that U-BOIN design is fit for purpose, a very powerful group. In that paper, they describe a plethora of methods how to satisfy requests of Project Optimus and to optimize dose findings. And they group those different methods in those that are all-in-one, continuous methods, and the two-stage methods. The two-stage methods would be what I showed you in the earlier slide. You do a dose escalation first, and then you do a dose optimization with 2 cohorts and randomize it. But there are methods where you sort of build that in, dose escalation, and the paper describes all of that. Now, while everybody thinks dose optimization can only occur in phase 1, and that is what Project Optimus asks, that is not quite true. The paper, the statistical paper, describes a number of methods where the dose optimization happens after phase 1, and they differentiate between those that have short-term endpoints, long-term endpoints, and if they have or not have a control group in the design. And you see those small differences already get you to 4 different design types. And then Dr. Ying Yan and colleagues finally have this paragraph. I love that one. Software. This group not only writes papers, they also produce extremely helpful softwares for the entire planet, free to be used. And they describe here in this paragraph how to get to it, so let's do that. This is the website, www.trialdesign.org. If you click into the web page, you see that there is a plethora of different study designs that you can click into, learn about them, and have helpful software with it. I recommend to click on filter by dose optimization, and then you only have 6 study designs left. These are the 6 study designs that you can get off the MD Anderson website for dose optimization study trial concepts. U-BOIN 1.2 is a further development of the classical U-BOIN design. It is a dose escalation rule, smarter, more complex than the 3+3 design, but it still follows the old concept of maximal tolerated dose. Find the maximal tolerated dose and then determine the recommended phase 2 dose from that. U-BOIN 1.2 goes a step further, follows the Project Optimus by including efficacy into the design. And tight U-BOIN 1.2 is a further development which allows including late onset toxicity and efficacy. U-BOIN is a two-stage design where, similar to one of my first slides of this video, you escalate first and then later you optimize. The two other ones here have this both together integrated in a one phase. The two next ones, the isotonic regression and the merit design, are both specifically designed to find the optimal biological dose. And they have both quite convenient software where you can actually type in how many patients you have now, and it tells you step by step, patient by patient, what to do. It also restricts your freedom. And droid design is something that came outside of oncology, where actually dose optimization is done more frequent and has more experience than we have in oncology. And they took a framework from there and turned it into oncology. It has the downside compared to the other five that the software isn't as convenient. You need to download the code. You need to have R on your computer to have this work. The other five work just by clicking into the website. This is what you see when you click in the BOIN 1.2 design in the MD Anderson website. You see they ask a couple of questions very similar to the original BOIN design, like number of doses, how many patients you have, and then, but the complex part is the relationship between toxicity and efficacy. Then once you made your decisions, it gives you tables like that. And in that table, they calculate something that they call the RDS. The higher the RDS, the better it is. And when the RDS is E, that means it is so undesirable that the cohort needs to be eliminated, not to be done anymore. It is somewhat in flow similar to the classical BOIN design, in thinking quite complex different. This is what you get when you click in the U-BOIN design software in this website. And you see this is a two-stage design. Stage one is quite similar to BOIN or 3+3, and stage two then starts looking at the things that Project Optimus really wants. In the entry, you have the similar questions like how many patients you have, and there is this utility trade-off between efficacy and safety. The isotonic regression design is the only one of the five designs where the authors are not MD Anderson people. This is a design that comes out of China. It is specifically designed to find the optimal biological dose. The questions in the beginning are kind of similar to what the other designs had. However, in addition to that, there is this part of the software here that once all the decisions are made and the study design is being defined, it helps you then conduct the trial. You can enter in how many patients had, how many toxicities, and it will give you step by step where you need to go next. And all the solutions I showed you up to now were for the classical situation that you have a new drug and you want to establish it in oncology. Dose escalate, find the efficacy. But obviously, drug development is more complex. Dr. Yap and his colleagues have looked into drug combinations in the setting of Project Optimus. Now they have a very long paper where they go through situation by situation and do that in great detail. But the total summary for me remains this sentence: no one-size-fits-all solutions. This is an example from the many study designs that Dr. Yap and his colleagues describe. So here they talk about something that looks very similar to a two-stage design, where there is a dose escalation for the monotherapy of drug 1 and then of drug 2, and then they test a whole bunch of combinations, and eventually they end up with 2 doses that get randomized and then tested in the typical way. So when I made this video, I called a lot of people and wrote a lot of emails, including FDA and colleagues in drug development and practicing physicians. And I collected what they said as recommendations how to handle the problems. Number 1, the problem of the sponsor. The clinical trial gets too large, we can't pay for it. Can be mitigated by early go, no-go decisions that are built in the clinical development plan and are sort of hidden within the protocol. yet they are early. They need to somehow show the sponsor that this molecule can be a drug, that it has some effect against cancer. That effect can be shown by monotherapy efficacy, tumor shrinkage, that's the classical one, but it could also be pharmacodynamic. You build that in early in the clinical trial. You tell the sponsors, if we don't have a go decision here, if we don't know that we have a drug, then we'll stop the whole thing. That can help you get started. Second point, and that's a point everybody made, was make use of the offer of multiple and early FDA interactions. Those guidelines are so non-specific, or no, they are specific, they're not so non-concrete. They say they depend on so many different things that you cannot really do this alone, and you don't really know what your contact partners in the FDA side think. So the best way is to go in early, describe what you want, listen to them, go back home, change what you have to do, and go back. And thereby slowly increase the complexity of your trial. With the first going there, be thorough, don't be sloppy with presenting the preclinical data. Preclinical data play a big role in the document for Project Optimus. And yet my recommendation would be in the beginning, keep the clinical component of the proposal simple. Just define the endpoints, how you want to compare the doses, and present the study design sort of framework naked, just the core, and then see what you need to add from their comments. It is not easy to determine the endpoints because the classical endpoints of numbers of dose-limiting toxicities for safety and tumor response for efficacy cannot give you answers to different dose levels with 20 patients only. That's impossible. That's why we have phase 3 studies. You need to be more specific and daring with the endpoints. For instance, neutrophil count. If that is a prominent endpoint, that is a quantitative endpoint and allows more statistical methods and smaller patient numbers to show differences between two different groups. Similarly, with response, you could substitute the response by a pharmacodynamic marker that is also quantitative, and then you can compare different cohorts with t-tests. Next recommendation. The elements that the FDA asks for being included, such as randomization, blinding, patient-reported outcomes, patient engagement things, don't add them immediately. Go with the naked protocol first and then see what they ask, and then you add everything that they ask. Pharmacodynamics plays a gigantic role in Project Optimus. And in some drugs and some cancers, this is really fantastic simple. You can take a biomarker that somewhat indicates the amount of cancer in the body, like PSA in prostate cancer. But in many cancers and for many drugs, there is no such assay. So if that's not the case, then the early development plan should include developing the PD assay. That is a recommendation from my friend Doug Feller. When you write the protocol, the old rule has been since a while already, write it relatively vague so that you have flexibility when you conduct it. And that is true for Project Optimus, including things like backfill cohorts. Don't make them mandatory. Write it in the way that you can use it when it comes about, but you don't have to. Then there is this problem of the clinicians that say, you are underdosing my patients. You can't do these protocol designs. There are too many patients underdosed. This is not right by the patients. That is a very valid concern. I have two answers. Neither of them really takes the concern away, but both of them make them a little better. Intra-patient dose escalation is specifically named in the document, and that can help. So if you have patients enrolled on a low dose, and then after 3, 5, 10 patients, you know that dose doesn't hurt anybody. And it's unclear if it helps, but the patients are still sticking around, maybe just because the cancer grows so slow, or because even that low dose did something. But by now, the next higher level also has data, and those data are also favorable for toxicity. So there is no reason to stay low. I'm recommending to allow intrapatient dose escalation. That can mitigate a little bit the underdosing problem during the escalation. And for the randomized part, where you have two doses that are randomized against each other, and you have to have safety and efficacy endpoints, where it's flipping between the cohorts is really difficult, screws up a lot of the outcomes. Futility rules can take place. Also a recommendation from my friend Doug Fuller. He said that was successful for him to prevent too many patients to be enrolled in a dose level that is too low. Design sharp futility rules, such as when that number of patients has been enrolled and there is no response, the dose level gets closed, or the other way around, too toxic. That's the classical and simple way of doing it. Those are the recommendations that I can come up with, and I hope they are helping. That's all I wanted to say about Project Optimus in stone cold information and opinions of others, ways how you can handle it. If you made it to that part of the video, maybe you want to hear my personal opinion. I have two opinions to add. One is there is no such thing as the dose for everybody. And the second one is, please loosen the handcuffs on our clinicians. They both go in the same direction. People differ. There are large people that have a large body and need more drug, and smaller people that need less drugs. There are bodies that tolerate more drug concentrations and others that tolerate less. There are cancers that need more drug. Even though they may have the same name as the neighbor's cancer, they both may be small cell lung cancer, but they still differ from each other. What we can provide in aggregated data in structured drug development is a framework of a drug, a starting dose of a drug. The drug should then be adapted, should be adjusted by well thought of dose modification guidelines that allow the practicing physician to adjust the dose to what is really necessary. Now that has become harder and harder for the practicing physicians because doses are described in the label and the insurances read those labels and the pharmacy of the hospital reads the labels, and then everybody thinks that is the one dose and one and only dose you can give, and when the clinician tries to do something else, they get into trouble. Please don't get them into trouble. Please help them find the right dose for an individual patient. And this is the end of the video. I want to thank a number of people that have helped me get the thoughts together and came up with recommendations what needs to be done. But this video is brought to you exclusively by Oncology Consulting Wolff LLC, which is solely responsible for the content. Pretty much all the mistakes that I made in this video are my mistakes. These wonderful people that have helped bringing the thoughts together have not reviewed or authorized co-authorship of that. Conflict of interest are not existing. Nobody paid for this video. No opinion was influenced by any kind of money. This is brought to you by this company, but that's the same as the author. I hope you learned something. I hope you enjoyed it, and we can see you again for the next videos. Bye-bye.